Chapter 21, Viruses, Lecture 21. Learning objectives. Viruses are acellular parasitic entities. The evolution of viruses is unknown. A virion consists of a nucleic acid core and an outer protein coating known as a capsid. There may also be other components, but this is the most basic. And then the Baltimore classification scheme is the one that's most commonly used to classify viruses, and it's just based on how mRNA is generated in each type of virus. So no one knows exactly when viruses emerged or from where they came, and this is because viruses don't leave fossils. Um, Basically, it's thought that modern viruses are a mosaic of bits and pieces of nucleic acids that are picked up from various sources along their evolutionary paths. Again, from the learning objectives, we know that viruses are acellular and they're parasitic. And because of the fact that they're acellular, they're not going to be found in any of the kingdoms because they are acellular, and all the organisms that are in the kingdoms are composed of cells. Now, unlike most living organisms, viruses cannot divide. What they do is they infect a host cell, and then they use the host cell to replicate identical virus progeny particles. Viruses will infect a diverse group of organisms, animals, plants, bacteria, and they exist somewhere between living and non-living. So living things grow, metabolize, reproduce. Viruses, again, they replicate, but we know that they have to use a host cell. They don't grow, they don't metabolize. Whenever they are replicated, they're replicated in their mature form. They are diverse. They will vary in their structure, replication methods, and then the type of host that they will target. So, something that targets E. coli may not target, it's not going to target a human because it's going to target E. coli. Nearly all forms of life have viruses that infect them. Animals, plants, fungi, bacteria, all of them have viruses that can infect them. And again, we know a lot about the evolutionary history of animals and plants and all that, but because of the fact that viruses don't leave fossils, very little is known about their evolution. Now, the discovery came, well, it's kind of interesting. This is kind of a two-part deal. So, there was this development of something called a porcelain filter, and it was called the Chamberlain-Pasteur filter. And what it could do is remove all bacteria that are visible in a light microscope from a liquid sample. And in 1866, this guy named Adolf Meyer demonstrated that a disease of tobacco plants, which is known as a tobacco mosaic disease, could be transferred from a diseased plant to a healthy plant or to a healthy one via liquid plant extracts. So what they did was they took diseased liquid extracts, put them through this filter, and the filter was unable to remove the disease, so it was able to be transferred. Now, the reason for this is obviously the filter wasn't able to filter out viruses which are small. So a virion, which is a single virus particle, is very small, 20 to 250 nanometers in diameter. Now, most viruses cannot be seen using a light microscope. It actually requires, well, it required the invention of the electron microscope, which didn't occur until the late 1930s. And that's whenever scientists were able to actually see viruses for the first time. Now, there are some viruses that can be seen with the light microscope, but for the most part, it requires an electron microscope. So the electron microscope allowed scientists to be able to see the viruses, see the morphology, the internal structure, how they differ structurally, and then started to use this to create a classification scheme. But first, we're going to talk about the evolution. So, uh, again, viruses don't fossilize, so we don't really know where they originated from. And there is not a single hypothesis that all scholars agree it explains the origin of viruses. There are three that are listed here. The de-evolution or regressive hypothesis, and this just has to do with, well, just suggests that viruses evolved from free-living cells. Then there's the escapist or progressive hypothesis that just suggests that viruses originated from RNA and DNA molecules that escaped from a host cell. And then there's a third hypothesis that is it discusses viruses evolving alongside of the cells that they rely upon as host. As far as the morphology, we already know that viruses are acellular, so they don't have the cellular structures like the organelles, ribosomes, plasma membrane. A virion, again, which is a single virus particle or molecule, I guess we'd say particle, not a molecule, is going to have a nucleic acid core, an outer protein coating or capsid, and then sometimes an outer envelope made of protein and phospholipid membranes derived from the host cell. Now, viruses can also contain other proteins like enzymes. And then the most obvious difference between members of viral families is the morphology, which is quite diverse. There are viruses, well, viruses come in many shapes and sizes, but they're consistent and distinct for each virus family. All virions have a nucleic acid genome covered by the protective proteins called the capsid, which we've already talked about. The capsid itself is made up of protein subunits called capsomeres. Now, some viral capsids are simple polyhedral spheres, while others are quite complex. Um, In general, the shapes of viruses are classified into four groups: filamentous, isometric, enveloped, and head and tail. So the filamentous are long and cylindrical. A lot of plant viruses are filamentous, including the tobacco mosaic virus. Isometric viruses are roughly spherical. Poliovirus would be an example. Envelope viruses have membranes surrounding capsids. This includes animal viruses like HIV. And then there's head and tail viruses that infect bacteria and have a head that is similar to a icosahedral virus and a tail shape like a filamentous virus. Many viruses use some sort of a glycoprotein to attach to their host cell via molecules on the cell called viral receptors. For these viruses, attachment is a requirement for later penetration of the cell membrane so they can complete their replication inside of the cell. So this is just showing the receptor and the actual virus particle binding. Um, this is getting more into viral morphology. So these are some of the more complex virions. The T4 bacteriophage, which infects E. coli. It has a tail structure. that the virus uses to attach to the host, and the head that houses the DNA. The adenovirus is non-enveloped. Well, it's a non-enveloped animal virus, which causes respiratory illnesses. What it does is it uses glycoprotein spikes protruding from its capsimeres to attach to host cells. Non-enveloped viruses can also, well, they also include polio, well, the viruses that cause polio, plantar warts, and hepatitis A. There's also envelope virions like HIV, and this is just showing the different morphologies. So this is the T4, yeah, it's a T4. This right here is the virus that causes respiratory illness, and then this is the one that causes AIDS. So, viral classifications. So first we can go through and we can read through this, which I'm not going to, because this actually starts to talk about how each of these viruses are transmitted and then what it causes. And we can look up here and see that we have rabies, which is shown, smallpox. So, but let's talk about classifications here. So, several classification systems have been used, and they've used it based on genetics, based on morphology. However, the classification system that's used most often today is the Baltimore classification scheme. And this is just based on how messenger RNA is generated in each particular type of virus. And we're not going to get into that in this particular lecture. Go ahead and read through that and read the different types of groups and how each virus in each of those groups is classified. So, in addition, viruses can be classified based on the design of their capsids. So, there are a number of classifications, and the type of genetic material and its structure is also used to classify virus core structures. So basically what this means is that there's a number of different ways that viruses can be classified. This right here is just showing different, these are just electron micrographs of various viruses that are showing the structure. So we see the capsid of poliovirus. So this right here is poliovirus. This is Epstein-Barr. This is mumps. This is the tobacco mosaic virus. And again, this is just talking about the capsid. So in this one, the capsid is naked and helical. And then finally, we have the virus that causes herpes. And in this one, the capsid is complex. So as a review, viruses are acellular parasitic entities. The evolution of viruses is unknown. A virion consists of a nucleic acid core and an outer protein coating, which is also known as a capsid. And then the Baltimore classification scheme is commonly used to classify viruses, and it's based on how mRNA is generated in each particular type of virus. And that is all I have for you for chapter 21.